Hola! Welcome to Chapter 6, Part 5, Microbial Nutrition and Growth. This is growth of populations of cells. So, the way that microbes grow, they use this process called binary fission. So it shows in the cartoon, here is a mother cell, mother microbe, with the cytoplasmic membrane, and then the cell wall is superficial to the cytoplasmic membrane. The chromosome replicates, and in this cartoon it takes about 30 minutes. Chromosome replicates, and then there's a septum, that's Latin for wall, that forms, and the septum is complete. And then you have two daughter cells, and then each one of those daughter cells replicates again. So from this daughter cell, you get two daughter cells, and from this daughter cell, you get two more daughter cells. And so the second generation will finish reproducing in 60 minutes, and then the third generation, 30 minutes later, the fourth generation, up to 120 minutes. So most unicellular microorganisms reproduce by this process of binary fission. And this is called the generation time. So the generation time is the time required for a cell to undergo one round of binary fission. So in this cartoon, the generation time of this microbe would be 30 minutes. In binary fission, in this first stage, the cell replicates its chromosome, shown here, which contains DNA, and proteins are synthesized. The cell then elongates, and you can see it gets longer here. It pushes the chromosomes apart. Remember, eukaryotic cells use mitosis, not binary fission. The cell then forms a new membrane and a new wall. And then this thing, the septum, or the division septum, separates the two daughter cells in the process called binary fission. And then the fourth stage, when the septum is completed, the cells, in this stage right here, they may separate, or here, it could be here or here. They may separate, or they could remain attached, depends on the microbe. Math and population growth. So growth of populations, microbial populations, is logarithmic. 2 cells divide into 4, 4 into 8, 8 into 16, 16 into 32, etc. So this means after, say, 7 hours of doubling, at a doubling or generational time of 20 minutes, you can have over 2 million cells from just one cell. So in 7 hours, you can go from 1 cell to 2 million cells. So the number of cells arising from a single cell is calculated by 2 to the n, where n is the number of generations that you have. So, and that's generation time. So, for example, the generation time in some cells is 20 minutes. So after 120 minutes, you'll have 6 generations of microbes. And then here's how you can calculate the total number of cells at any given time. Just know that the calculations can be calculated. You can read over this if you like, but there will be no generational time math on the exam. So the generation time is also called the doubling time. It's the time required, like we said, for a cell to grow and divide from an initial or starting population. So the starting population could be just 1 cell, or it could be 10 to the 6th or 1 million cells. It depends on the starting population number. The generation time depends on the environmental conditions. So under optimal conditions, so they have the right temperature, the right pH, they have enough food. The average bacteria have a generation time of about 20 minutes. So a small number of microbes can cause food poisoning in a very short time. Most bacteria can require a generation time of 1 to 3 hours. Some may need days or even weeks. Treponema pallidum that causes syphilis takes weeks. Growth is logarithmic, as we said, not arithmetic. So like we said, 2 cells divide into 4 cells, 4 cells into 16, 16 into 32, etc. So from starting time of 0 to 2 hours, you go from 1 cell to this many cells. If growth were arithmetic, you'd have a really low number of microbes, which might be good in some cases, but microbial growth is not arithmetic. So in arithmetic growth, you'd go from 1 cell to 2 cells to 3 to 4 to 5 to 6. So at the end of 2 hours, you'd only have 8 cells. Scientific notation, you've probably heard of that before. So for example, 1 times 10 to the 12th is a trillion. And bacteria grow into trillions of cells, so scientists have to use scientific notation. So consider a species. It divides every 20 minutes, so it has a generational time of 20 minutes. After 1 hour, which is 60 minutes, it divides every 20 minutes, that's 2, 4, 6. You'll have 3 generations. After 10 hours, that's 600 minutes, you'll have 30 generations. So after 10 hours, and starting with only a single cell, after 10 hours, you could have 2 to the 30th, or, well, this is the form you use, 2 to the 30th. So you'd have 1 billion number of cells. It's better to express it as 1.07 times 10 to the 9th, or 1.07 billion cells in 10 hours. So growth curves. Let's look at the y-axis. This is a number of cells. It's a log scale on the y-axis, and then time is just regular, not a log scale. And these are the phases of microbial growth. So when you take your microbe of interest, let's say you introduce it into a new culture and you're trying to grow it. There'll be a lag phase here. The cells won't increase. Then they'll grow logarithmically or exponentially. And then they'll hit the stationary phase where they won't grow anymore. And then if you don't change the media, they'll go into the death phase. So a growth curve is just a log plot of the number of cells in a growing population over time. So here again, on this side, letter A is an arithmetic scale. Letter B is a logarithmic scale to count the number of cells. And you can see why you can't use an arithmetic scale, because you can't accommodate the graph on a single population, on a single page, as the population grows. So population growth would go up here, and you couldn't get the numbers on a single page. So the solution is use a log scale. So you can accommodate small numbers at the lower end and upper end of the graph. So let's talk about the phases. The lag phase of microbial growth. The cells are metabolically active, but they're not dividing. They're in the lag phase. So you took them from their home broth and you scooped them up in your loop and you inoculated them into a new medium. They just need to adjust to that new medium. They don't begin reproducing right away. They synthesize enzymes, they make their ribosomes, etc. And this lag phase can last from less than an hour to several days. And then you go into the log phase of growth. You get rapid logarithmic growth because the conditions are optimal. That means there's a lot of nutrients in the broth, and there's less number of waste products in the broth. DNA and protein synthesis is maximized, but the interesting part is when they're dividing, they're most susceptible to antimicrobial drugs. And also, When you are going to stain for a cell, gram stain, you want to get the cells in the log phase. That's when they stain the best. Stationary phase. The stationary phase, the nutrients are kind of decreasing, the wastes are starting to build up, the reproductive rate is going down relative to the log phase. But the rates of cell production and cell death are approximately equal during this stationary phase. You can postpone the stationary phase of your culture, and definitely using this guy over here called a chemostat. So a chemostat will, this overflow tube, you will continuously remove the waste, the old media and the waste, and then this flow regulator here will allow fresh media to come into the chemostat. So you could keep your cultures going for a long time with the chemostat. The death phase is the last phase of microbial growth. It's logarithmic as well. It's the log phase. Cells are dying faster than they are produced. So that's the end. The death phase, the end.